Ever wondered where the word muscle actually comes from?
I sure I have.
It's straight from the Latin mus, meaning little mouse.
Mm-hmm.
Kind of playful, really, like how flexing muscles look. Sort of like tiny mice scurrying just under your skin.
Huh. Little mouse. I like that.
But muscles are, so much more than just, say, biceps. We're talking about nearly half your body's mass. They dominate the heart, shape the walls of our hollow organs. It's huge.
Absolutely massive.
So today, we're embarking on a deep dive into this incredible world. Muscles and muscle tissue, essentially. Chapter 9, from the 10th edition of Human Anatomy and Physiology.
Okay.
Our mission. To really get how our bodies convert chemical energy, ATP, into, well, directed mechanical energy, movement.
And our aim for you, listening in, is to unpack the really crucial anatomy and physiology here.
Right.
We want to offer clarity, accuracy, no glossing over details, but connecting it all to the real world. Clinical stuff, too. We'll go from tiny cell motors all the way up to, say, powering a marathon.
Sounds good. So let's start big picture.
Yeah.
The three main types of muscle tissue. Each one's a specialist.
Okay.
First up, the one we usually think of, skeletal muscle. These are packaged into your actual muscles, the ones attached to bones.
Got it. Biceps, triceps, quads.
Exactly. Longest cells in the body, and they have these obvious stripes, striations. Crucially, they're voluntary.
Meaning you control them consciously.
They decide, jump out of the way, look left.
Yeah.
You tell them what to do. They contract super fast, powerfully.
But they tire out?
Right. They tire relatively easily.
Right.
That sprint finish, you feel it afterwards.
Yeah, that fatigue is interesting. It's like a built-in feature for those quick, powerful bursts.
Then there's cardiac muscle. Name gives it away, right?
Only in the heart.
Only in the heart. It's striated, too, like skeletal muscle. But here's the key difference.
Where?
Totally involuntary.
You don't think beep, beep, eat?
Nope. It contracts on its own rhythm, set by the heart's pacemaker. Now, your nervous system can definitely speed it up, if you're running.
Or see that last slice of pizza, maybe?
Yeah. Yeah, exactly. And finally, smooth muscle. This is in the walls of hollow organs. Think stomach, bladder, airways.
The internal plumbing, kind of?
Pretty much.
Yeah.
These cells are elongated, but no striations, and like cardiac, totally involuntary.
Right.
Their contractions are different, too. Slow, sustained. They just keep working, pushing fluids, food, whatever, through those internal channels.
It's amazing how these three, despite being so different, share these four core characteristics that let them do their jobs.
Okay, what are those?
First, excitability or responsiveness. Think of muscle cells as listening. They can react to a signal, usually a chemical, by changing their electrical state. Flipping a switch, basically.
Getting ready for action.
Exactly. Second, contractility. This is the big one. The unique ability to shorten forcibly when they get that signal. That's muscle's signature move.
Nothing else does that quite the same way.
Nope. Third, extensibility. They can stretch even beyond their resting length when relaxed. And fourth, elasticity. That ability to recoil, snap back to their resting length after being stretched. It's like a built-in shock absorber. Keeps them safe.
Excitability, contractility, extensibility, elasticity. Got it. And those enable the main functions, right? Like the most obvious one. Produce movement, locomotion, grabbing things, even just moving your eyes. All muscle.
Right.
Then maintain posture and body position. This one's wild. Even when you feel still, your muscles are constantly making tiny adjustments against gravity nonstop.
You just collapse otherwise.
Totally. They also stabilize joints. While they're pulling on bones to move you, they're also holding those joints together, making them stronger.
Dual role there.
Yeah.
Yeah.
And finally, they generate heat. It's a byproduct of contraction, but it's vital for keeping your body temperature stable.
And smooth muscle has a few extra tricks up its sleeve, doesn't it?
Oh, yeah.
Well, it forms valves, regulating flow in your digestive tract, urinary tract. It controls your pupils dilating and constricting. And those erector pili muscles, goosebumps, that's smooth muscle too.
Wow. Okay. So let's really zoom in now. Skeletal muscle, the gross anatomy, peeling back the layer. Take a whole muscle, like your bicep. That's an organ. It's wrapped in this tough outer layer, the epimysium.
Epi, meaning upon.
Right. Now, inside that, the muscle fibers aren't just loose, they're bundled together, like bundles of sticks. These are fascicles.
Fascicles, got it.
And each fascicle is wrapped in its own layer, the perimysium. Peri meaning around.
Makes sense.
Then finally, you get down to the individual muscle fiber, which is actually a single long cell. And that's wrapped in a really delicate sheath, the endomysium. Endo meaning within.
And what's clever is how these layers connect, right?
Exactly.
These sheaths, the epi, peri, and endomysium, they're all continuous. They merge together at the ends and blend into the tendons.
So the course gets transmitted.
Precisely. The fibrous pull, the sheaths pull, the tendon pulls, the bone moves. Plus, they provide elasticity and pathways for nerves and blood vessels. Super important.
Speaking of nerves and blood, each muscle gets its own nerve, artery, and usually one or more veins.
Dedicated supply lines.
And here's a key thing for skeletal muscle. Every single fiber has its own nerve ending controlling it.
Wow, that allows for really fine control.
Incredible precision. And the blood supply is intense. Muscles are energy hogs when they work, so they need constant oxygen and nutrients and efficient waste removal.
Absolutely critical. Which brings us to attachments. How do they connect to bone?
We talk about origin and insertion. The origin is usually the anchor point, the bone that doesn't move much.
Okay.
The insertion is on the bone that does move. When the muscle contracts, insertion moves towards the origin.
Makes sense. Pulling action.
Attachments can be direct, where the epimysium just fuses right onto the bone, covering the periosum.
Like it's glued on.
Sort of, but more common is indirect. The connective tissue wrappings extend beyond the muscle as either a rope-like tendon or a sheet-like aponeurosis.
Tendons are what we usually picture.
Yeah. And they're tough. They can handle rubbing against bone, and they save space compared to fleshy muscle crossing a joint. Very efficient design.
Okay, structure makes sense. Shall we go smaller? Inside the fiber.
Let's do it. Inside the skeletal muscle fiber. It's this huge cylindrical cell, multiple nuclei, just under the surface membrane, the sarcolemma.
Multinucleated. That's unusual for cells.
It is. And the cytoplasm, the sarcoplasm, is packed. Glycosomes for glycogen fuel and myoglobin.
Like hemoglobin, but for muscle.
Exactly. Stores oxygen right there in the muscle cell, ready to go.
Clever.
Now, inside, 3 highly modified structures are key for contraction. First, myofibrils. Hundreds of thousands of these rod-like things fill up like 80% of the cell volume.
Wow, packed tight.
Totally. And these are what give the muscle fiber its stripes, the A bands and I bands. They're the contractile engines.
And these myofibrils are made of repeating units.
The sarcomeres, right? The smallest functional unit. From one Z disc to the next Z disc, like little boxcars lined up end to end down the whole myofibril.
The fundamental contractile machine.
And inside the sarcomere, that's where we find the actual proteins doing the work, the myofilaments.
Okay, the real engine parts.
Yep. You've got thick filaments made mostly of myosin. Each myosin molecule has this tail and 2 heads.
Heads are important, right?
Crucial. They form cross bridges. They grab onto the thin filaments to generate force. They're staggered along the thick filament, except right in the middle, the H zone.
Got it. Myosin heads do the pulling.
Then you have the thin filaments, mostly actin. Actin has the binding sites where those myosin heads attach. But it's not just actin.
Regulatory proteins, too.
Exactly. Tropomyosin. It spirals around actin, and when the muscle's relaxed, it physically blocks the myosin binding sites, like a guard rope.
Okay, keeps things turned off.
And then troponin, a complex of 3 proteins. One part binds to actin, one binds to tropomyosin, and the critical one binds to calcium ions.
Ah, calcium.
The trigger.
The trigger. When calcium binds troponin, troponin changes shape and yanks tropomyosin out of the way. Binding sites exposed.
Okay, that's the on switch. There's also elastic filaments made of titin. They run from the Z disc to the thick filament, helping the sarcomere spring back after stretching. And we should mention dystrophin too, linking thin filaments to the sarcolemma. Important later.
It's incredible organization. And these tiny proteins interact with another key structure, the sarcoplasmic reticulum, the SR.
Right, the SR.
It's basically a specialized, smooth ER that wraps around each myofibril like a lacy sleeve. Its main job, store calcium and release it on demand.
That calcium reservoir.
Precisely. That release is the final go signal for contraction.
And how does the signal get from the surface deep inside to the SR?
Ah, that's where the T-tubules come
Those are
in.
tubules, okay.
Those are deep tunnels and vaginations of the sarcolemma diving right into the cell at each band junction. They carry the electrical signal, the action potential, deep inside.
Like express tunnels for the signal.
Exactly. Ensures every sarcomere gets the message almost instantly, so the whole fiber contracts together.
Super fast communication.
And where a T-tubule runs between 2 enlarged ends of the SR, the terminal cisterns, that forms a triad.
Okay.
At the triad, proteins link up. Voltage sensors on the T-tubule detect the action potential. They change shape and trigger gated channels on the SR to open.
And calcium floods out.
Calcium floods into the cytosol. That's the crucial link. Electrical signal triggers calcium release. excitation-contraction coupling.
So we have all the players. How does the muscle actually shorten? The sliding filament model.
That's the one. It's elegant, really. During contraction, the thin filaments slide past the thick ones. Overlap increases.
But the filaments themselves don't shrink.
No, crucially, neither thick nor thin filaments change length. They just slide relative to each other.
Okay. How does the sliding happen?
The myosin heads are the key. They act like tiny ratchets. They bind to actin, pivot. That's the power stroke, pulling the thin filament towards the center of the circumere.
Pulling from both ends towards the middle.
Exactly. As this happens all along the myofibril, the I bands shorten, the Z discs get closer together, the H zone disappears. But the A band stays the same width.
Because the thick filaments aren't changing length.
Right. And this coordinated pulling shortens the whole muscle fiber. Rigor mortis is a really stark example of this needing ATP.
Ah, yes. Stiffness after death.
After death, ATP production stops. Without ATP, the myosin heads can't detach from actin. They get stuck in that cross bridge.
Irreversibly linked.
Until the muscle proteins themselves break down. It perfectly shows that detachment is an ATP-driven step.
Fascinating. Grim, but fascinating.
Okay.
So how does the fiber know when to start this whole process? Needs a signal, right?
It absolutely does. It all starts with a nerve impulse arriving at the neuromuscular junction, the NMJ.
The meeting point, right.
Where the end of a motor neuron axon gets really close to the muscle fiber's sarcolemma, there's a tiny gap, the synaptic cleft.
Okay.
The axon terminal has vesicles, little sacs, full of the neurotransmitter acetylcholine,
ASHA. The chemical messenger.
Yep. Nerve impulse arrives, ACH gets released, diffuses across that tiny cleft, and binds to ACE receptors on the muscle fiber side.
Like a key fitting into a lock.
Perfect analogy. And that binding opens ion channels. More sodium ions, Na+, rush in than potassium ions, K+, leave.
Creates an electrical change.
Exactly. A local depolarization called the end plate potential. If it's strong enough to hit threshold, boom. Action potential ignited in the sarcolemma.
The go signal spreads.
And just as quickly, an enzyme in the cleft, acetylcholinesterase, breaks down the ATPase, stops the signal, prevents constant contraction. Very precise timing.
So problems here would cause issues.
Big time. Think about myasthenia gravis. It's often an autoimmune disease where the body attacks its own ACE-sec receptor.
So the signal can't get through properly.
Right. Leads to muscle weakness, drooping eyelids. It really highlights how vital that specific NMJ connection is.
Okay, so the action potential is triggered. How does it spread and reset?
Well, at rest, the sarcolemma is polarized, negative inside, positive outside. A-chain binding opens channels. Na+ rushes in, causing that local depolarization, the end plate potential.
Okay.
That local change triggers nearby voltage-gated Na+ channels to open. More Na+ rushes in, causing more depolarization. It becomes a wave, the action potential, spreading across the entire sarcolemma.
Like dominoes falling.
Good analogy. Then, repolarization happens almost immediately behind it. Na+ channels close, voltage-gated K+ channels open, K+ rushes out, restoring the negative charge inside.
Resetting the system.
Exactly. And there's a brief refractory period right after where it can't be stimulated again immediately. Ensures the signal travels in one direction.
So electrical wave spreads. Then
Right.
what? Excitation, contraction, coupling.
Right. The action potential dives down those T-tubules deep into the cell.
Okay.
As it travels, it triggers those voltage-sensitive proteins in the T-tubule membrane. They change shape.
And talk to the SR.
They signal the SR to release its stored calcium. A huge flood of K2+ pours into the cytosol surrounding the myofibrils.
And that calcium is the final trigger for...
the cross-bridge cycle.
Let's walk through that cycle. Calcium's out.
Calcium binds to troponin, remember?
The gatekeeper protein on the thin filament.
Right. Calcium binding makes troponin change shape. It pulls tropomyosin away from the myosin binding sites on actin.
Sites exposed.
Now, the energized myosin heads can latch onto actin. That's cross-bridge formation.
Okay. Attached.
Then the power stroke. The myosin head pivots, pulling the thin filament toward the sarcomere center. ADP and phosphate are released here.
Generated the force. Now what?
A new ATP molecule binds to the myosin head. This causes it to detach from actin. Critical step.
Needs ATP to let go, like rigor mortis showed.
Exactly. Then that ATP is hydrolyzed, split into ADP and phosphate. The energy release recocks the myosin head into its high energy position, ready for another cycle.
And it repeats as long as.
As long as calcium levels are high enough in the cytosol and ATP is available. Attach, pull, detach, recock, over and over, pulling those thin filaments inwards.
An incredible molecular machine.
Yeah.
So that's at the fiber level. How does this translate to lifting a weight or just holding something?
Good question. We need to talk about muscle tension versus load. Tension is the force the muscle exerts. Load is the opposing force, the weight of the object.
Okay. Tension versus load.
And contractions fall into two main types based on what happens with length and tension. First, isotonic contractions.
Isotonic. Same tension.
Yeah.
Not quite right.
Yeah, the name's a bit misleading. It means the muscle length changes to move a load. Tension usually exceeds the load here, two subtypes.
Right. Concentric.
Muscle shortens, does work, like lifting that book, curling a dumbbell.
And eccentric.
Muscle generates force as it lengthens, like slowly lowering that dumbbell or walking downhill. Interestingly, these are more forceful, maybe 50% more, and often cause more delayed soreness.
The negative part of a lift.
Exactly. The other main type is isometric contraction.
Isometric.
Same length.
This time the name fits. Tension builds, maybe even peaks. But the muscle length doesn't change. The load is greater than the force the muscle can generate.
You're pushing against a wall or trying to lift that piano.
Perfect examples.
Yeah.
Or just holding your posture. Isometric contractions are vital for stability.
How does the nervous system control how much force a whole muscle produces?
Through the motor unit, remember that's one motor neuron and all the muscle fibers it connects to.
Okay. One neuron, multiple fibers.
Right. And the size varies. Muscles for fine control, like eyes, fingers, have small motor units, one neuron, maybe just a few fibers.
Precision.
Exactly. Big weight-bearing muscles, like your quads, have large motor units. One neuron controlling hundreds, even thousands of fibers. Right. And when that one neuron fires, all the fibers in its unit contract together. All or none for the unit.
So if you stimulate just one motor unit briefly, you get
a muscle twitch. A single quick contraction and relaxation. You can see it on a myogram.
It has phases. 3 phases. The latent period stuff's happening internally, EC coupling, but no tension yet. Then the period of contraction cross bridges cycle, tension rises. Finally, the period of relaxation, calcium goes back into the SR, tension falls. Relaxation is usually a bit slower than contraction.
But we don't move in twitches. Our movements are smooth.
Right. We need graded muscle responses. Varying the strength. 2 main ways the nervous system does this.
Okay.
First, changing stimulus frequency, firing the neuron faster. If signals arrive before the muscle fully relaxes, the contractions summate. Wave summation.
Piggybacking contractions.
Kind of leads to stronger tension. If it gets fast enough, you get a quivering state, unfused tetanus. Even faster, a smooth, sustained maximal contraction, fused tetanus, though that leads to fatigue eventually.
Okay, frequency matters. What's the second way?
Changing stimulus strength. This is recruitment, or multiple motor unit summation. Basically, activate more motor units.
Bring more fibers into the game.
Exactly. A weak signal activates just a few small units. A stronger signal recruits more and larger units. And this follows the size principle.
Size principle.
Yeah, it's really neat. The smallest, most excitable motor units are recruited first. Then, as more force is needed, progressively larger, less excitable units are added.
So you get fine control for light tasks and ramp up the power smoothly for heavy tasks.
Precisely. Very efficient.
And even when relaxed, muscles aren't totally slack. Right. Muscle tone.
Right. Even at rest, spinal reflexes are constantly activating a few motor units here and there, asynchronously. Doesn't cause movement, but keeps the muscle firm, ready to respond. Good for posture, joint stability too.
Okay. Muscles need energy, ATP. But they only store a tiny bit. How do they keep going?
They have 3 main ways to regenerate ATP on the fly.
3 pathways, okay.
The quickest is direct phosphorylation. Uses creatine phosphate, CP. CP stores a high-energy phosphate, quickly gives it to ADP to make ATP.
Instant power.
Almost. Lasts about 15 seconds. Think 100-meter dash weightlifting rep. Very fast, but very limited supply.
Okay, 15 seconds isn't long. What's next?
The anaerobic pathway. Glycolysis. Breaks down glucose without oxygen.
Anaerobic is no oxygen.
Right. It's faster than aerobic, but less efficient. Yields only 2 ATP per glucose molecule. And produces lactic acid as a byproduct.
Ah, lactic acid. That burn.
Yeah, contributes to fatigue and soreness. This pathway can power activity for maybe 30-40 seconds, up to a minute. Think a slightly longer sprint, intense burst.
Still pretty short term. What about endurance?
That's where aerobic respiration comes in. Takes place in the mitochondria. Needs oxygen.
Okay.
It completely breaks down glucose, fatty acids, amino acids. Yields a ton of ATP, around 32 per glucose. Much slower process, but sustainable for hours.
Marathon running fuel.
Exactly. So the body shifts between these systems depending on the intensity and duration. Short burst. CP. intense few minutes. Anaerobic kicks in hard, long duration. Aerobic takes over.
Makes sense. What about muscle fatigue, just running out of ATP?
Not usually, no. It's more complex. It's a physiological inability to contract despite the signal still coming. Often involves ionic imbalances like potassium accumulating outside the cell or phosphate buildup inside interfering with calcium release, or cross-bridge function disrupts that EC coupling.
So the communication breaks down, essentially.
Often, yes. The specific cause can depend on the type and intensity of exercise. And after you stop, you need EPOC, excess post-exercise oxygen consumption, often called the oxygen debt. It's the extra oxygen you breathe afterwards to restore everything.
Paying back what you used.
And more. Replenish oxygen reserves, convert lactic acid back, restore glycogen, resynthesize ATP and CP. Resetting the system takes time.
Okay, so how much force can a muscle generate? What affects that?
Several things influence the force of contraction. 1, number of fibers recruited. More units, more force.
Simple enough.
2, size of the fibers. Bigger fibers, like from resistance training, hypertrophy, generate more force.
Makes sense.
3, frequency of stimulation. As we said, summation and tetanus increase force dramatically.
Right.
And 4, degree of muscle stretch. There is an optimal length, the length-tension relationship, where filaments overlap perfectly for maximum cross-bridge formation. Too stretched or too shortened, force stalks off.
Interesting. Optimal overlap. And what about speed and duration?
Velocity and duration depend mainly on muscle fiber type, the load itself, heavier load equals slower contraction, and recruitment patterns.
Fiber types. Let's get into those 3 main ones.
Yep. Slow oxidative fibers, SO. They contract slowly, use aerobic respiration mainly, hence oxidative. Lots of myoglobin, so they're red. Very fatigue resistant.
Endurance fibers.
Exactly. Think marathon runners. Postural muscles. They're thinner, less powerful individually, but can go for ages.
Okay.
Next.
Fast glycolytic fibers, FG. Contract rapidly, rely on anaerobic glycolysis, glycolytic. Low myoglobin, so they look white. Powerful, large diameter.
But they tire quickly.
Very quickly. Fatigue easily. Ideal for short, intense bursts. Hitting a baseball, sprinting, heavy lifting. Power fibers. And the third?
Fast oxidative fibers, FO or FOG. Kind of intermediate. Contract fast like FG fibers, but use aerobic respiration like SO fibers, oxidative. Have decent myoglobin, moderate fatigue resistance.
The all-rounders.
Sort of. Good for activities needing speed and some endurance, like walking or sprinting longer distances. Most muscles have a mix of all 3.
And the mix can change.
To some extent, yes. Genetics plays a big role in your baseline mix, which is why some people excel naturally at endurance, others at power. But training can shift the characteristics.
Ah, how exercise changes things. The overload principle.
Right. You have to challenge the muscles. Aerobic exercise, jogging, swimming increases capillaries, mitochondria, myoglobin, makes muscles more efficient, fatigue resistant, can even convert some FG fibers towards FO characteristics.
Better endurance?
Yep. Resistance exercise weights mainly causes hypertrophy. The fibers, especially FG fibers, get bigger. More myofilaments, more glycogen stores, increases strength and power, can shift some FO towards FG.
Building size and strength?
Exactly, but what happens if you don't use them?
Disuse atrophy.
Yeah. Immobilize a muscle, like in a cast, and it weakens fast, maybe 5% strength loss per day. If nerve supply is lost long-term, muscle tissue can actually be replaced by fibrous connective tissue. Irreversible sometimes. Use it or lose it really applies here.
Okay, that covers skeletal muscle pretty well. What about smooth muscle? The smooth operator.
Right. Involuntary, non-striated, walls of hollow organs.
Structurally different too.
Very. Cells are spindle-shaped, single nucleus, no coarse connective tissue sheaths, just endomysium, often arranged in 2 layers in organs, longitudinal and circular.
Why 2 layers?
They work together for peristalsis. Circular layer constricts the tube, longitudinal layer shortens it. Alternating contractions propel substances along, like squeezing toothpaste from a tube.
Ah, clever. Microscopic differences.
Lots. Innervated by the autonomic nervous system, often via varicosities that release neurotransmitters diffusely over multiple cells. Less direct than the NMJ. SR is less developed, no T-tubules. Instead, caveolae in the sarcolemma trap extracellular calcium. No sarcomeres, so no striations. Myofilaments arranged diagonally, no troponin. Calmodulin is the calcium-binding protein.
Calmodulin instead of troponin. Big difference.
Huge difference in the activation mechanism. Contraction is still sliding filament, calcium-triggered, ATP-powered, but the sequence is different.
How does it work?
Calcium enters, mainly from outside, binds to calmodulin. This activates an enzyme, myosin light chain kinase. MLCK. MLCK then phosphorylates the myosin heads, activating them so they can bind actin. Much slower process than in skeletal muscle.
Slower, but efficient.
Incredibly energy efficient,
Right.
can maintain tension for long periods with very little ATP use. That's key for smooth muscle tone, maintaining vessel diameter, organ pressure, etc. And contractions are often synchronized via gap junctions between cells. Whole sheets contract together.
Regulation is complex, too.
Very. Nerves, hormones, local chemicals, all can influence smooth muscle. And a neurotransmitter might excite one smooth muscle type but inhibit another, depending on the receptors.
And that amazing stretchiness.
The stress relaxation response. It can be stretched a lot, then adapts to the new length and relaxes, but can still contract. Allows organs like the stomach and bladder to fill up without triggering immediate emptying. Really vital.
Two main types.
Mostly unitary smooth muscle, or visceral. Cells coupled by DAP junctions contract as a unit, found in most hollow organs. Then multi-unit smooth muscle. Fibers are independent, no gap junctions, need direct nerve stimulation. Found in large airways, arteries, eye muscles, erector pili. Finer control.
Okay, almost there. Development and aging.
Muscles develop from embryonic myoblasts. Skeletal fibers form by fusion of many myoblasts, hence multinucleation.
Regeneration.
Skeletal muscle has limited regeneration via satellite cells. Cardiac muscle, very little, mostly scar tissue. Smooth muscle regenerates pretty well. Cells can divide.
Sex differences.
Men generally have more muscle mass due to testosterone, but strength per unit mass is similar.
And things that can go wrong, like anabolic steroids.
Yeah, synthetic testosterone variants can increase mass, but the side effects are severe and often permanent. Liver damage, heart issues, hormonal chaos, mood swings, roid rage. Not worth the risk. Even things sold as enhancers can be harmful.
And inherited conditions. Muscular dystrophy.
A group of diseases where muscles degenerate. Duchenne muscular dystrophy, DMD, is the most common, serious form. Sex-linked, affects boys mostly. What causes it? A defective gene for dystrophin, that protein linking filaments to the sarcolemma. Without it, the membrane tears easily during contraction, calcium leaks in, damages fibers. They eventually die and get replaced by connective tissue. Progressive weakness.
Tragic. And aging. Muscles just decline.
There are age-related changes. More connective tissue, fewer muscle fibers, gradual mass loss, sarcopenia, but
Exercise helps.
massively. Regular exercise, especially resistance training, can significantly slow down or offset sarcopenia, maintaining strength and function much later in life.
Finally, muscles don't work alone, do they?
Not at all. Constant homeostatic interrelationships. Skeletal system provides levers. Nervous system controls. Cardiovascular delivers fuel, removes waste. Skin helps regulate heat. It's all connected.
Oh, what a journey. From that little mouse idea to actin and myosin dancing, ATP fueling everything, how we adapt. It's just incredible how these tissues let us move, stand tall, even control things inside without us thinking.
Absolutely. And considering all that complexity, that adaptability, makes you wonder, doesn't it, about the potential, the sheer marvel of your own body's physiology working constantly.
It really does. Well, thank you for joining us on this exploration today. We truly appreciate you being a part of our last-minute lecture family.
Always a pleasure.
Until next time, keep flexing those knowledge muscles.